Welcome back, Life Sciences. As I said during a break, it's always necessary just to get the stand up, do a little bit of wiggle, get the oxygen going, get the blood flowing, right? Otherwise, we just get so bogged down and none of that information actually goes into our head. So what have we looked at? We just did a brief recap on all the microorganisms that we have looked at. And now we're going to look at a few questions that we could ask you on these different kind of sections. Right, so let's start off with our very first question. I always like to start off with a bit of an easier question. When it comes to the microorganisms, you'll see, guys, pictures and information you're going to need to read. Okay, we have 3 diagrams here, so let's go. It says study the diagrams. We've got 3, and I'm hoping just by looking at them, you already can identify which they are. And they say to you they are all microorganisms, and then we're obviously going to answer questions on that. I've kept the picture at the bottom here so that we can just refer to it. It says name the group of microorganisms. That is basically the ones that we looked at, the kingdoms. So it was a virus, bacteria, protist, or fungi. That's what we're looking at of B and C. So if we have a look at B, we have a look here. What do we notice? We can see this characteristic DNA in the middle, and it's got a layer around it. So a B is going to be a virus. And C, right, what do we look at C? We see it's got these, looks like little, right, threads, and they've got these big, right, dark balls on the top, and those balls are going to carry spores. And the only one that has spores over here that we can see, that was our fungi group. Nice, easy question to start off with. Now, studying question, provide labels for 1. Number 1 is our bacterium, and that is a flagella or a flagellum. You can use either 2. Right, and that is the tail. Try not to use tail because that's not the correct one. Number 2, as we said, this is a virus. So let's just put here bacteria. All right, and we said this is fungi. Right, what was number 2? Number 2, I'm going to put here a nucleic acid. Because remember we said a virus can have a nucleic acid, and that can either be DNA or RNA. So I would have accepted either of those answers. Number 3 is that black ball that I see to you on top. It's a really dark color. That's what we see. And it holds spores, so we call it a sporangium. Right, if we go back to all our distinguishing characteristics, remember what I said, you had to know what a simple, typical diagram of each of the microorganisms would be. Okay, now the next one, give the letter. Guys, not the word, not the name, give the letter. Please read your questions properly. It's even in capital letters to tell you, which is not considered to be living. So out of the 3, which one did we say was acellular? They only asked you for the letter. That would be a nice big A. Is eukaryotic. Now, if we have a look, this is non-living, so it's not a cell. Bacteria are prokaryotic, which only leaves us with number C. Fungi are eukaryotic. So that is by a process of elimination. Now again, name the shape of the bacterium in diagram A. Guys, so if, nice little clue here, so if you didn't know it was a bacteria, they actually gave you the answer. And remember, bacteria have shapes. This is a rod-like one, and we call it a bacillus, or a rod-shaped bacteria. But we do prefer if you were able, to give its scientific name. Okay, now the next questions that we're going to start off, very often with microorganisms, we're looking at case studies, looking at giving us data, etc., reading from graphs, reading from tables. It's sometimes nice and easy because the answer is there. You just need to be able to read carefully and look carefully. Okay, let's have a look at the question. Right, whooping cough, and if you have never heard of it before, that is okay, because remember, what does it tell you? It's a bacterial disease. Okay, so that's really important. It's telling you it's a bacterial disease that affects some infants. I actually had it when I was a child. I can remember I was off school for quite a while because I got it. An investigation was conducted to determine, so as soon as we see to determine, that's the aim of our experiment. The relationship between vaccinating infants against whooping cough and the cases of whooping cough reported. Okay, so basically what you're looking for here is you're looking at the concept vaccinations. All right, that's what we're looking at. Do vaccinations have an effect on getting the bacterial disease? Now, when we looked at, the functions, remember we said a vaccination is there to help our body cope when we get a disease, so that we might not get it as in such a serious way than if we didn't have the vaccines. Okay, so now the data relating was collected a long time ago, right, between 1965 and 1996. That's fine. We just need to be able to read from a graph. Okay, what did they look at? The following data was collected. Now, guys, this is quite unusual because you got a graph that's got two sets of axes. Have a look here. You've got one on one side over here. I'm going to change the pen color quickly. On the side over here, the number of cases reported. So have a look here on this part of the graph, all right? Number of cases reported is over here. Then on the other side, now I'm going to change back to blue, the percentage of infants that were vaccinated. They're on that side. So you've actually got two y-axes. The graph below shows the data collected. So let's see, all right, we're going to come back to this graph quite often. Let's see what kind of questions they're going to ask. This is a very common question when it comes to microorganisms, because we can use microorganisms for experiments. So the concept of a dependent and independent variable, right, is going to be quite important. Now, the first one, dependent. A dependent variable is the results. And our results, right, are always going to be on the y-axis. Now, why did I say this is quite interesting is that for the first time, you've actually got two dependent variables. Now, those dependent variables are the following. Okay, guys, so as I said, we have two. So if we look at number one, I'm going to put number one on the side here, and I'm not going to write it all out, but we're going to write the whole description. Right, let me do it, so otherwise, because I can't write on this thing. Number of, all right, let me go back, let me do that, of cases of whooping cough reported. All right, there we go. What's the next one? Number two. We're going to write that out just at the top there. Percentage, I'm going to just do it nice, I'm going to cheat. Percentage of infants vaccinated, all right, against, and I'm going to just put WC for whooping cough. Right, so we have two independent variables. We're looking for two results. How many of the infants were vaccinated and how many had the cases of whooping cough? What was my independent variable? My independent variable is on my x-axis. All right, remember, I lie, all right? And that's exactly what the x-axis is down. It's downward. And that would be the year. Right, we decided on which years we were going to collect this data. We had no choice. Remember, we decide, I decide. We have no control over the results. Okay, guys, now the next question is you need to have a look at both of the graphs. It says, state the conclusion based on the data shown. Now, remember, what were we looking at? Is there a pattern between vaccination and children getting sick. Now, if we have a look at our graph, what do we notice? When our vaccinations are down, right, if you have a look over here, if our vaccinations are down, what happens after that? Our cases increase. So what is our conclusion? What is it all about? Is there a relationship between vaccination and children getting sick? And it definitely is. The conclusion is the more children that were vaccinated, there we go, or you can say it the other way around, right? The least, or the number of cases of whooping cough, right, decreased. So we saw, what can we conclude? That vaccinations help prevent the whooping cough, right? So that's what I noticed. Looked at our trends. When one went down, when the cases of children went up, it's because the number of children were not being vaccinated. Okay, our next question, right, is again is an experiment. Let's see what Liso did. Liso carried out an experiment to find out what factors, right, would help mold to grow on bread. She took 4 slices of white bread, remember, all the same bread, and put different substances, this is what I'm choosing, on each slice of bread as follows. So on slice 1, she left it dry. On slice 2, she put some tap water on. On slice 3, she put some lemon juice. And slice 4, she put some sugar water. Guys, I hope you notice she used the same amount. Controlled variable. Controlled variable. Okay, fixed so that you make sure that it's reliable. Okay, each slice of bread was put in a Ziploc bag and kept in a dark cupboard. All right, so remember, mold is fungus. What conditions do fungus grow best in? Warm and dark and moist. That's why she put it in the Ziploc bag to keep all the moisture in. Right? And she left it at a temperature of 25 degrees for 2 weeks. She observed the growth of mold on the 4 slices of bread and worked out the percentage area of the bread covered by mold. The results are shown in the graph below. Okay, so if we have a look here, a nice picture of the different slices of bread. Okay, state a hypothesis for this investigation. Now guys, I like to start off, I teach my kids to start off a hypothesis, which is a prediction, with the following words. It is expected that. Right, because that kind of puts them in the right frame of mind to make a prediction. Now, tell me, it is predicted that. Look at the information. The bread with sugar water will have the most mold. Look, I used my information because what did it show me? It showed me the sugar water. So I'm being lucky here. I'm saying, yes, I can see the results. It is expected that the bread with sugar water, there's my one variable, will have the most mold. There we go. Independent variable, and there is my dependent variable. Nice marks, 3 marks for my hypothesis if I was going to give you the marks. The independent variable, what did I choose? It's always on, right, the x-axis. And what did I choose? I chose what substances to put on the bread. That's what I chose. All right, what substances must I put on the bread? Explain why mold did not grow on the bread sprinkled with lemon juice. Now we're going to have a flashback to maybe when you did biochemistry. What does lemon juice do? It changes the pH, right? And what does the change in pH mean? It's going to change how enzymes work. And here you're going to see, explain why mold did not grow. The lemon juice interfered, I'm going to use the word, interfered, all right, with how the bread mold, its enzymes, this is a difficult question, all right, could break down the glucose in the bread. Right, so guys, if the bread mold couldn't break down, the enzyme couldn't break down, right, so what happened was the mold couldn't grow because it wasn't getting enough food. This is it's a difficult question. It's a higher order question. All right, the second last question was, why was dry bread included in the experiment? Guys, why? It's a control. That's why. We always got to make sure we have a control. So we wanted to see what would make it, so we took out, all right, no substances. So to make it see, all right, what it was. We always have to have a control. Take away something to make sure what is going to grow there. All right, identify and briefly describe the type of nutrition of Rhizopus. Guys, it's saprophytic. What does that mean? Right, it needs to grow, it needs the hyphae to penetrate or to grow into the bread to get, right, to get the glucose in the bread. See, it's heterotrophic. Another word there you might have used, it is heterotrophic, right? It cannot make its own food, so it needs to be able, right, to get its own food. Okay, guys, that's all we have. Just for now, we're going to take a quick break and we'll be right back.